My name is David Rempel. I'm a faculty in the Department of Bioengineering at the University of California at Berkeley. The experiment you're going to see involves stretching tendons, and really the purpose is to try to understand the underlying mechanism to tendon disorders at the wrist and elbow. These tendon disorders are common among athletes and workers who do repetitive work with their hands. Krishna Sundi, one of the doctoral students in our laboratory, will demonstrate several features of the system, specifically the measurement of the cross-sectional area of the tendon. He'll load the tendons into the actuators. He'll demonstrate the system loading the tendon cyclically over a 24-hour period, and then remove the tendon from the system.
Okay, so this is the loading system that we've designed and built. As you can see, it consists of 6 actuators. These are high-precision actuators which are capable of applying displacements of 1 micrometer. In series with the actuator is a load cell to record loads applied to tendons. In series with the load cell are these clamps which extend away far enough so that they can be submerged in media in order to keep tendons viable. This entire system would then be placed in a water bath to maintain the media at 37 degrees Celsius. What I'm going to show you right now is how I would go about loading a tendon into this system. The first step we need to do is measure the cross-sectional area of the tendon so that we can apply the appropriate stresses. Pass me the tube with the tendon. Now, these tendons have been harvested from a New Zealand white rabbit. They're just been brought over, so they're still fresh, within less than 2 hours of the rabbit being euthanized. The portion of the tendon